So we're now going to turn to think a little bit more deeply about transgenerational epigenetic inheritance. So we've spent some time thinking about the environment, with then considering whether or not these environmental influences might be transgenerationally inherited. But now we're going to think about transgenerational inheritance that's not influenced by the environment, but rather is due to epigenetic variability that's stochastic, that occurs naturally. So again, it's nice to think about examples, non-mammalian examples. And the best example of transgenerational epigenetic inheritance in plants is that in Linaria vulgaris. So this is a plant that has two different habits, two different types of flowers, that was first described back in 1744 by Linnaeus, and that's who named the plant. So it has these two versions of the flower, the wild type flower, which has bilateral symmetry, which I think you can see best in this picture here, this bilateral symmetry of the flower. But the other version of the flower, which is called the peloric mutant, has quite a different flower structure. You can see that here. It has a radial symmetry, and this radial symmetry you can see with the small petals coming out is quite different to the wild type flower. So for a very long time, these were believed to be two different strains of the Linaria vulgaris plant. However, it was in 1999 that a publication came out saying that you can't find any difference genetically from what they looked at between these two flower types, but rather they found that there was a heritable DNA methylation difference, and this was at the L6 gene. So this heritable DNA methylation difference had apparently been ongoing and occurring with a very high penetrance, being passed on generation by generation since 1744 at least, when Linnaeus described these two flower types, and probably even before that. And so now this is believed to be a very good example of epigenetic inheritance in the plant, although their reprogramming happens in a different way. And this is really an epimutation, if you like, that is heritable. So the extra evidence for it being an epimutation is that sometimes if you follow the inheritance of these peloric mutants, then you'll get a rarer version and get a branch of a plant that comes out having wild-type flower habit, in the rest of the plant having these peloric mutant flowers. And this is extra evidence that it's indeed epigenetic, because epigenetic changes are reversible, whereas if it was a mutation, it would be extremely unlikely and probably impossible to have some reversion of that mutation happening. So this is interesting in the context of a plant, but let's think about mammalian examples in more detail. So the first time that a mammalian example of transgenerational epigenetic inheritance via the gametes was described was actually for a transgene. So I'm showing you a large pedigree here that describes the expression of a transgene. So a transgene being a gene that we've actually added in to this mouse strain ourselves. It's a gene that's not normally found there, and it was found particularly so for when you add a gene in that's of bacterial origin. So it's extremely foreign, if you like, because it's nothing that's even mammalian derived or even looks like anything mammalian, but rather is one that we would normally find in bacteria, and so therefore a mammalian cell can detect that it's not quite right being there. So in this first generation, I'm showing an animal that expresses that transgene, so it's active, a male mated with a wild-type female. From this point onwards in the pedigree, I'm not showing you the wild-type offspring because they're not relevant, so I'm just shrinking down the pedigree to make it a bit smaller by excluding these wild types. But if we look at the transgenic offspring, they come in two varieties. They can either express the transgene, male or female, like these ones shown here, like their parent did, and so I'm showing them as red, or they can be silent for the transgene. And in that, what I mean by silent for the transgene is that they do not express the transgene. It's epigenetically inactivated. So if you then follow the inheritance of the expression state from these parents that have the silent transgene, we know that if you follow the father that had the silent transgene, he can still produce offspring through the next generation, and then the generation after that can have an active transgene. So while it's been silenced in this animal, it can be reactivated again when it goes through the germline. If we look at the inheritance from an active mother, she can produce active or silent offspring. So they can either express the transgene, so it's red, or not express the transgene, and so it's silent in this case. The difference occurs when you take a female that has the silent transgene, so she's not expressing the transgene. And from that point onwards, it doesn't matter how many generations you follow, this transgene will never be reactivated again. So this transgene, once it is silenced through the maternal germline, now no longer can be reactivated. There's no genetic difference at this transgene. It's exactly the same sequence as the one that her brother got, or her other sister. This female just happened to inherit it in the silent state, and this can no longer be reactivated. And this suggests it's not going through those rounds of epigenetic reprogramming to be able to clear the epigenetic marks off between generations. It's escaping those rounds. Now, I'd like you to consider how this differs from genomic imprinting that we thought about in week 4. So in genomic imprinting, we know that the state that's set up in the germ cells is then perpetuated for the lifetime of the subsequent generation. But that's quite different to what we're seeing here. And that's because here, although we have it persisting, it is never ever reset again. Whereas for genomic imprinting, it gets reset every generation. It's reset at every time they go through primordial germ cell development. So it would be reactivated again each generation. And that's not what's seen here for this pedigree here following female transmission, this inheritance of that silent state. So we also know that it happens at other alleles that we'll talk about in later lectures. But I'd like to first think about what this really means and what it means and how we can test for it. So I started talking about in the previous lecture about how we can think about transgenerational epigenetic inheritance happening in humans. But because this is such a controversial topic, we really need to have the appropriate controls to be able to say for sure that something is transgenerational epigenetic inheritance. So the sorts of things we want to think about if it is transgenerational epigenetic inheritance via the gametes is, when is the phenotype established? So this is something that I thought about and discussed with regards to the maternal care. We could take pups and cross-foster them over to a separate mother and find that it was actually the mothering style which altered the epigenetic state in the hippocampus in the offspring. So this is the phenotype being established post-birth as opposed to earlier. These experiments can't really be done in humans. We can also examine, what we also need to know is there some effect of the uterine environment? One argument would be that in the case of the transgene, the mother who had a silent transgene may have somehow a different uterine environment to a mother who had an active transgene, and it could be this that was setting up this perpetual silent state from that point onwards. We also know that if the phenotype is established late, like for the hippocampus, the maternal care and the stress response, then if you cross-foster, then you can take into account whether or not this is being established, but also we can take into account parental genetics. So in other words, whether it was actually an underlying genetic difference in the parents which allowed for the establishment of these different phenotypes. So the point I want to make here is that all of these sorts of questions which we really need to address to talk about transgenerational epigenetic inheritance via the gametes are really best addressed in either mouse or rat studies, or in a model system that we can control the environment for and where we can perform these studies in the laboratory. So while we really are interested not in whether this occurs in rat or mouse, we're much more interested in whether it happens in humans. We can only really ask the questions appropriately in rat or mouse. So the question then becomes, I think, why should we discriminate between something that's an environmental effect or some other effect which is not transgenerational epigenetic inheritance via the gametes, and something that is genuinely fitting with this definition of transgenerational epigenetic inheritance via the gametes? And I think the reason that we care is because it has we can interpret this information differently. So we can interpret it differently, and we can also act differently in terms of public health. So if we want to think about how this is happening in terms of mechanism, we need to know whether it's something that's happening in the gametes or something that's happening much later on. That also means we need to think about whether or not it's when the gametes are developing that is the most sensitive period, or whether it might be some later time point in development. At the moment, we know that there's a lot of information that pregnant mothers or mothers that are planning to become pregnant receive about what their diet should be. And so we need to know, is that the appropriate information, or should they also be very aware that in the months prior to falling pregnant, they also need to take care with their diet, as is actually advertised right now. They should avoid alcohol for this whole period. These sorts of pieces of information only come about if we have the very specific information about these periods of exposure and about the periods when it might be relevant. And the difference between transgenerational epigenetic inheritance via the gametes and something that's established much later mean that we can discriminate between when the most critical periods are in development, and therefore we can give the best advice in terms of health. So in the next lecture, we're going to go into detail about one, perhaps the best example of transgenerational epigenetic inheritance via the gametes in the mouse. This is the agouti viable yellow allele.